Now we're going to practice a little bit with karyotypes and gathering information from a karyotype. Again, a karyotype is the arrangement of those chromosomes. So using the diagram below, answer the corresponding questions. The following diagram is a set of human chromosomes. This diagram is known as a karyotype. There are several things that one can see in such a diagram. You will discover that these things through the following reading questions. Okay, notice this is karyotype A that we're looking at, and we have pairs 1 through 22 that are numbered 1 through 22, and then we have the XY down here. This would be our sex chromosomes. This determines the gender of the individual. This one is the XX, 2 chromosomes at X, nothing at Y. So this would be a female. Notice that there's 2 chromosome 1, 2 chromosome 2s, but notice that they are not attached to sister chromatids. So apparently this sample was taken maybe during anaphase as they had pulled apart from the centromeres. So we have just one chromatid there, one chromatid there, one chromosome, just one copy. Sometimes we see them as X's, where this would be a chromatid, and then it would have a copy over here attached to it. It would be the sister chromatid. But then notice that these are singles. These are singles, not even little X's. So this would be one individual chromosome, maybe the one from mom, and one individual chromosome, maybe from dad. And this one may be the one from mom and the one from dad, and so on. Okay. So, let's see. It says, karyotype A is the first set of chromosomes that we'll look at. Notice that the chromosomes are all paired up. How many pairs do we have? Well, we have 1 through 22 that's numbered, and then our pair of sex chromosomes down here, so there are 23 pairs. Change color so we will see those answers. 23 pair for a total of 46. And look closer, you'll see that the pairs of chromosomes are paired according to certain likenesses among those chromosomes. Okay, remember we talked about homologous chromosomes? We were talking about meiosis, how they pair up in homologous chromosomes. Well, notice they're about the same size. That one's about the same size as that one. That one's about the same size as that one. Even though they're curled up, they're about the same size. And notice these bands. This is called a banding pattern. They stain the chromosomes, and the stain attaches at certain bases, certain proteins within those chromosomes. So they have a similar banding pattern, which also means that they have the same genes in the same places. So they're arranged according to size and banding pattern. So they are arranged according to size and banding pattern. Which would also indicate the same genes in the same places. Each karyotype chart also shows some important details about the sex of the organism. Look at the particular karyotype on the chart and what is the sex of the organism. Well, we just said XY is that's the sex chromosomes. That's what determines the gender of this individual. And there are 2 chromosomes at chromosome X, nothing at the Y. So if there's nothing at the Y, this is a female, a girl. Number 4 says the chromosomes in the karyotype also show some light and dark bands. What do you think these regions of light and dark bands are? Okay, well, these are banding patterns that stained at particular protein or at particular bases. And they can also indicate where certain genes are located. If you start at chromosome number 1 and go through chromosome number 22, you will see a trend in size correlation. What is that trend? Well, notice chromosome number 1 is quite large. 1, 2, and 3, they're quite large. Until we get down to here to 22, that is really small. So the trend is they're arranged from largest to smallest. Again, the X and Y is going to be a little different because they're just put at the end anyway. But 1 through 22, they're arranged from largest to smallest. And how do you think this trend affects the number of genes present in a particular chromosome? Well, we would expect there's room for more base pairs in those longer chromosomes, so we would expect them to have more genes. So we would expect there to be more genes in the longer chromosomes. Number 7 says each pair of chromosomes are numbered. How many pairs of numbered chromosomes do you see? Well, we have 1 through 22. So there are 22 pairs of numbered chromosomes, and then our 23rd pair is labeled as XY. So there are 22 numbered pairs. And what are the numbered pairs called? We call those autosomes. If it says it's autosomal dominant or autosomal recessive, that means it is on the autosome. It's not on the X or Y. We talked about sex-linked traits. Those are on the X and Y. And if we say it's an autosomal trait or it's an autosomal dominant or autosomal recessive, that means it's located on one of the other chromosomes, not on the X or Y. So 1 through 22, those are known as autosomes. Autosomal chromosomes. There's a final set of paired chromosomes that are called the sex chromosomes. What are the possibilities for the sex chromosomes? Well, if it's a female, it could be an XX. And if it's a male, it would be an XY. And number 10 says you'll notice that the pair of chromosomes are characterized into groups. These groups are lettered A to G. Can you see any reason or trend in the categorization of these chromosomes? If so, what is the trend? So if we go back and look, this right here, 1, 2, and 3 make up group A. And notice they're about the same size. That line goes through their centromeres. And centromeres are where they would have been attached to the sister chromatids if they had their sister chromatids here. They did not. But if they did, they would be attached at the centromere. The centromere divides them into what we call the P arm and the Q arm. P being the short arm here, it kind of looks like in the middle almost. Right here it's a little more dramatic that this P arm right here would be tiny, and then the Q arm would be longer. Okay. So they are grouped according to size. These are about the same size, and then there's a big change in size from there to here. And also according to the location of the centromere. You notice this is group C down here. They're all about the same size. And the centromere is kind of in the same place. Notice these have very short little P arms, almost no P arm, in this group D. So they are grouped according to size and location of those centromeres. So those groups are based on size. So the groups according to similar size and centromere location. Number 11 says every karyotype is specially named. The naming system is based on 3 things. One is the number of chromosomes, such as 46. 2 is the sex of the organism, XX or XY. And 3 is any extra chromosomes that would cause a genetic defect, such as plus 18 if it were trisomy 18. So what would we expect this one to be named? And this one we just have 2 in each pair like we would expect. So we have 22 pair, that would be 44, 45, 46. There's 46 chromosomes. It is an XX female, and there are no extra or missing chromosomes in there. So we would name this one 46 XX, and then nothing there. Okay. Now karyotype B, can we notice a difference here? These are paired up with their sister chromatids. This is maybe from metaphase or late prophase, where they've already replicated, they're condensed into their little X's that we can see. But this would be one chromatid, and this would be the copy chromatid of that chromosome from maybe mom. And this chromosome number 1 maybe from dad. That would be the original chromosome, and that would be the copy, and they're held together with their centromeres. So that's a little different from up here. Up here in karyotype A, they were not paired up with their sister chromatids. This is probably in anaphase after they've pulled apart from each other. And in this one, it would be probably metaphase. This is where they harvested these chromosomes. And notice as we're looking through here, we would expect to find 2 in each pair, 2 pair in 1 through 22. And when so far so good. When we get down here to 21, we see a difference. They should be pairs. There's 2 there, 2 there, 2 there, and so on. At 21, chromosome 21, we have 1, 2, 3 chromosome 21s. This would be referred as trisomy, tri meaning 3. This would be trisomy 21, also known as Down syndrome. So this would be a disorder. This individual, instead of having the normal 46 chromosomes, would have 47. We have an X and we have a Y. Because there's a Y here, this would be a male. This would be a male with trisomy 21. So how many pairs of autosomes does this karyotype have? Again, it still has what we would expect, the 22 pairs of autosomes. 22 pairs of autosomal chromosomes. Except 23 is not a pair. This time it's a triplet. It would be trisomy 21. How many pairs of sex chromosomes do we have? There's just the 1 pair of sex chromosomes. In this case, it would be an X and a Y. This will be an XY individual, making it a male. If you look closely at the chromosomes in karyotype B versus A, there's a big difference. Can we tell what it is? And it gave us a hint that it happened at prophase 1, and that is that they are attached to their sister chromatids. So the sister chromatids are still attached, so they're like little X's. Sister chromatids are still attached together, so they appear as little X's. Look at all the pairs. Do we see a strange circumstance? And what is that special circumstance? Well, we just talked about that. We're moving along. We have 2, 2, 2, all of them like we would expect. And then we get to our chromosome 21. And we have 1, 2, 3 of these teeny tiny little chromosome 21s. So, we find 3 chromosome 21s. And this would be Down syndrome. Or also known as trisomy 21. This one we would name, this individual has an extra chromosome, so it would be a 47. It's a male, XY, and plus 21. So try the rest of those on your own and see what you can do with the other karyotypes.